So the first lecture, I probably won't go the whole time, unless you want me to drone on for 45 minutes. Okay. So the first thing we're going to do is we're going to look at an overview of evolutionary biology. And what we're really going to try to do today, my main goal is to really introduce you to kind of evolutionary thinking and the basic process of natural selection and the various kinds of data that we can use. And I'll show you some examples, and we'll leave you with some examples of some thought questions and things how we might approach questions in evolutionary biology. And there's a second chapter. I don't require you to read it, but it's kind of the history of evolutionary thinking, predating Darwin, through Darwin, and up to the present. It's kind of an interesting chapter. It's not going to be a requirement, and I'm not going to lecture on it. So basically, as you think about this, when we think about, if we try to distill everything that Darwin did into 2 key points, which is extremely difficult to do, his revolutionary insights I think can be condensed down into these 2 parts, which is the current diversity of life that we see, right? And this has to do with the phylogenetic diversity in particular groups of organisms, all the unique traits that are associated with them, all the adaptations that we see of them. He basically, his key revolutionary insight is that current diversity of life that we see on Earth is descended from previously existing species, and that many of these current species share a common ancestor with these previously existing species. Some of these existing species have gone extinct, right? Some have been maintained throughout and coexist with the current species. But understanding this notion of common ancestry and diversification through time it was one of his really key components. And he used a lot of different pieces of evidence to support that. And again, this comes back to the idea that I'm not the sort of person that's going to tell you have to believe in evolution, right? And I'm an open-minded guy. I'm not, but what I'm here to present in this course is basically how we approach the diversification of life through evolution via natural selection. And most scientists believe there's a preponderance of evidence that natural selection shaped diversification on Earth. Many other evolutionary mechanisms shaped diversification of life on Earth besides natural selection. And part of what we're going to try to do is show you how that occurs. And the other thing that's really important is we are documenting natural selection occurring in populations. Thousands of them are being documented every year. So it's easy to document natural selection, and we can extend that over time to lead us to believe that there's a preponderance of evidence that natural selection has led to the diversification of life on Earth. Okay. One of the things we're going to talk about is looking at the present forms of current species. And when we talk about forms, right, what we're talking about, what are the functions, traits, features, and/or adaptations that organisms have and species have that have been the result of natural selection. And we'll talk about what natural selection is. So, and this is not to say that everything that we see on Earth in the diversification or the things we see on Earth are all derived from natural selection. There are a lot of evolutionary processes that influence natural selection. This is part of what we'll be spending the first third of the course in population genetics. And the second part of the course is on things besides natural selection that influence evolution. Can you guys think of do you remember anything from biodiversity? What are some other processes that affect evolution and diversification besides natural selection? Yeah, mutation, right? That's the original source of genetic variation. We'll be coming to that. Can you think of anything else that's not so predictable and, Yeah, artificial selection is we're going to be talking about kind of a form of natural selection that we impose on things we're interested. Anything else? Genetic drift, okay. That's always the big mystery to everybody. We'll be spending 45 minutes on this sort of random, what we call stochastic process that leads to populations becoming different from one another. And we think it's a very important thing in terms of nucleotide or sequence diversity that we see. Okay, so the key component of today's lecture is understanding the process of natural selection, how we examine it experimentally and through observation to test hypotheses. So what I will try to do in each lecture, I'm going to give you kind of what I think are the key points at the beginning of each lecture, and then sometimes I lose my steam towards the end of the semester, okay, but I'll try to be better. And then at the end, I try to reinforce what I think are the key concepts and the key take-home messages, right? And as you study and you start thinking about for exams and looking through materials, kind of focus on those key concepts and kind of the data and experiments that tested them, okay? So what kind of data do we use? Well, it is incredible the amount of data and kinds of data that biologists and evolutionary biologists and population geneticists use, ranging from fossil data in A to B with either gelada baboons, I think, in Africa. I think they're in Africa. I'm not sure. But these would be like what's that guy doing? What do you think he's doing? What kind of, he's peering through. Does he really need binoculars? Unless he's extremely, he has some sight problems. Okay. But this is an example of somebody who's capturing behavioral data, right? Looking at the social interactions between this, probably an extended family of baboons, and how mother and son, mother and daughter, father and offspring are interacting with one another. And a lot of time, behavioral observations are a key component of data that we can collect. C is a group of, what do those look like? Those look like butterfly wings. Would you believe me if I told you those are all from a single species in the Amazonia? Pretty remarkable, huh? And you're probably saying to yourself, how can that be one species? Well, we'll try to address that question, okay? So what you can see there is an incredible what do you see? Diversification in terms of the size and spots on the wings, right? The coloration of the wings. So again, these are all, as we go throughout, and I'm just introducing you to a lot of different, how we approach these questions. But one of the things we might ask ourselves is what sort of led to that diversification and that divergence? And it turns out these are populations of the same species that are separated and isolated from one another, and they've diverged in terms of these wing colors and these wing patterns, okay? And we might ask ourselves what might be leading to that. And if you had to fathom a guess, what do you think? Yeah, and this is one of the key components that we're looking at is in evolutionary biology, population genetics, it's always an interaction between organisms and their environment that lead to natural selection. And it might be that certain kinds of traits or these phenotypes might be favored under different kinds of environmental circumstances. Now the environment that's a pretty broad term, isn't it? So what we're going to be spending a lot of time on is how do you characterize the environment? We can characterize the environment in terms of the physical environment, right? Such as temperature, humidity, rain, precipitation, water availability, all these sorts of things. And we can also think about it in terms of what we call the biotic environment, right? Those butterflies, who do you think they are potentially interacting with in terms of other species that might influence those patterns? Give me two guesses. Huh? Yep, birds might be one, right? So they would be the prey, and you'd have a predator. That particular kinds of wing patterns might influence their survivability, right? Whether they're camouflaged or not in a certain kind of environment, okay? What else do you think color is important for, not only in terms of a predator, but in what other kind of social context do you think might be important? Yep. So reproduction is obviously going to be another key component, and the diversification of a lot of things that we see in terms of coloration, magnitude of traits, often have to do with traits that are involved in reproduction and mate choice, okay? So we're going to be spending a lot of time looking at these sorts of things to see that sort of thing. There we have sort of developmental patterns. One of the things that is really important right now that's going on, at least in evolutionary biology, is what's called evo-devo. It's evolutionary development. How do we understand the genesis, angiogenesis, and the genes that are triggered during this is embryonic development, but it can also be through juvenile development up to adulthood. How do developmental patterns differ between individuals of the same species in different populations? And how does gene expression change as a function of environment? So we'll be looking at some of those things too. And this is amazing. We're still showing this in a textbook. Do you have any idea what that is? That's what I used to do in 1998. Huh? Yeah, nope.
Is that when people would like write out, like the genes?
the sequence data. So that is an old radiograph of how we used to do DNA sequencing, okay? So, and my PhD graduate students and I would sit there. We do it independently, make sure we got the right thing. But of course now this is all automated through what are called sequencers and things like that. So we don't do that anymore. But one of the biggest sources of data right now, and we'll be doing this a lot, is the availability of DNA sequence data to reconstruct phylogenetic histories of organisms and all sorts of things to personalize medicine. There's a lot of stuff going on in molecular biology in terms of DNA sequence data. Okay, so we have lots of data. This is really hard to read. This is what I hate. Okay, so one of the key features of Darwin's work was on the role of domestication and artificial selection in the divergence of organisms. And as you know, humans like certain things, don't we? And all of those have come about through domestication of ancestral wild species. And what this is a map of is fairly straightforward. Here's years before present. Here are the various regions. A is Southwest Asia, South Asia, East Asia, and then here we have South America, Mesoamericana, and then North America. A whole series of crops and animals that were domesticated, right? Oftentimes when we look at, what we have is a long history of domestication. There was often a long period of what we call pre-domestication, okay? So if you were to go into Mesoamericana in Mexico, what is one of the key crops that originated in Mexico that is an important component of our diet? Corn, okay? So we know very well the ancestral species from which all of the various forms of corn came from. It's called teosinte, okay? And if you go into, primarily it's in southern Mexico, Oaxaca, western Mexico, this is where the ancestral species that was domesticated gave rise to corn. Do you think it looks like corn? It looks extremely different. If you were to compare that to a piece of sweet corn or any sort of corn, they have tiny little kernels. They are tiny little fruits, tiny little stems, and you think they're sweet and succulent? No, they're hard as can be. They'll kill you. No, they won't kill you, but you have to boil them for a long, long time to soften them, okay? So all of these things, there are a whole list of from wheat to barley to lentils and all these sorts of things. All of these things have ancestral species in the wild from which they went through often a fairly long period in blue, which is pre-domestication use. Humans used them. They really didn't undergo artificial selection and domestication, but then there's often then a longer period of domestication that's highlighted in red and all those sorts of things, okay? So one of the really fascinating things is looking at domesticated plants, the genetic changes that have occurred, what's happened to the traits and all these sorts of things. And how did humans domesticate both plants and animals? Well, we took them out of, we basically took them and we basically, not as sophisticated as what we do now, but went through a process of artificial selection, right? And if you think about it, what is artificial selection? Yeah. So what artificial selection usually is, it's usually humans select for usually a very narrow range of desirable traits, often just kind of one, okay? And what's the process of artificial selection? How might it be? Well, we'll talk a little bit about how it's related to natural selection, but basically what it is artificial selection results in what we call phenotypic change. That's not an unusual term, is it to you, phenotype? Okay. So artificial selection results in phenotypic change in a population. I'll come back to this last question as I go through this, and I'm going to ask you a question on how might other traits change. So what's this domesticated plant that we have down there? Strawberries, okay. And have you ever eaten a wild strawberry? Huh? They don't taste good. Okay. Well, I think they don't taste good to our palate. Okay, because what do we like in strawberries? What do you guys like in strawberries? Do you like tartness, acidity? You like sweetness and juiciness, right? So that's basically large. So basically what artificial selection does in most domesticated crops is sort of, in general, larger components to the things we're interested in and a certain flavor profile, okay? And as we look at this, what happens in artificial selection is really analogous to natural selection. Certain kinds of individuals are allowed to reproduce, right, and found the next generation. We do that by selecting those individuals. In this particular case, plant seeds from the plants that produce the largest, juiciest fruits. You do that over generation after generation, and assuming certain things, what's eventually going to happen is you're going to lead to the evolution of larger and juicier fruits, okay? So you harvest the food, you only select those individuals, you do this for tens, hundreds of generations. This phenotypic change can occur really rapidly depending on the genetic component, right, and how strong you select for large, juicy fruits. And when you think about it, what do you think when you do domestication, when you think about the distribution of, say, size and juiciness, right, on the x-axis, right? What do you think it looks like in kind of the original population? Do you think it looks like a kind of a normal distribution or a bell-shaped distribution where small, non-juicy ones are at the tail? Most of them are kind of intermediate, and then you have some way out on the right-hand tail of the distribution that are extremely large and juicy, right? Those are the ones you select. And you keep selecting that tail of that distribution for generation after generation, right? And what it results in is that phenotypic change over time to the desirable trait. Okay, so and Darwin wrote a whole book on all the examples of artificial selection through domestication that resulted. And anytime you look at those ancestral species in that previous graph compared to the thing we have right now, oftentimes you wouldn't even know they were closely related. And in fact, it took a lot of evolutionary molecular analysis to find what teosinte was, and that in fact was the ancestor to corn. So, okay. So what do you think, if I were to ask you, so you've selected for juiciness and sweetness, right? Do you think some other traits might have changed in plants, in these strawberries, or even just plants in general? What do you think's happening when you select for large, juicy fruits in terms of other traits that a plant has? And what's it? So strawberry is a fruit, right? Huh? Well, what is it? It's a rose. Okay. Okay. So it's a rose. Well, it is a rose. Okay. But let's think about these other components of the plant, which are, let's look at the above ground part, the leaves, right? Stems, roots, right? What do you think, let me ask you, how do you think the leaves of the ancestral plant differ from the leaves of a strawberry plant? In the ancestral one? Yeah. That's in general what you often see. There's often a trade-off. And what do you think? I was going to try to focus in on not only just the size and number, but what do you think about the composition of those leaves? So when you think about strawberries are gardened, they're farmed, do you think they have a lot of pests and insects wanting to eat them? But what do? We try to reduce the damage from insects through it can be organic means, it doesn't matter, but it can also be from inorganic means and insecticides and things like that. But do you think the pressure from insect pests in general is greater in the ancestral population than the domesticated? Well, it turns out it is. Yeah. So it turns out when you go in and look at a lot of pre-domesticated plants, and when you look at leaves, it turns out not only if you were to eat them, guess what would happen? You'd probably want to barf. Okay? Because they have actually what are called a lot of secondary chemical compounds that are actually toxic to the things that eat them. So they invest a lot of energy and resources into the production of defensive compounds. And that often might come at some expense to the fruit, right? You think about an organism only has so much, so many resources to invest in, right? So it turns out a lot of wild plants tend to invest a lot more in sort of preventing insect damage and things like that, and it might come at some cost to also the size and juiciness of the fruit. Okay? So again, had you ever thought of that? Okay. So I'm not saying you have to memorize it. This is a lot of what we do in evolutionary biology. You can select for traits, but there are also consequences on other features of organisms as you undergo this selection process. So. Huh? Yeah. And what it turns out, probably what happens is, domesticated strawberries right now in these large farms, right? If you didn't provide a lot of, they would be extremely prone to insects eating them. Okay, if they didn't have any protection from the things we provide them, they don't have a lot of those chemical defenses that prevent insect. Yeah. I don't know. Potato blight. Anybody know? I'm not sure I know. I think it was a. I don't know. I have to go back and look. So. But anyway, the key point here is organisms have to deal with a lot of different things, and we have to think about them kind of in their totality. Okay, so here are the key conditions for artificial selection and natural selection. We'll be coming back to this in the next lecture and really spending a lot of time looking at it. But what we say here is there are three basic components. I think you probably all did you all have this in biodiversity at least, where these three basic components of natural selection that are necessary conditions for natural selection to occur in a population. And what we mean by natural selection occurring or artificial selection is the change in the phenotype over time. So to get that change over time, you need variation in traits or a trait among individuals within a population. So in that strawberry example, in the pre-domesticated, there had to be variation in fruit size. Okay. We also assume that variation is heritable. We'll be spending a lot of time on that in terms of genes that influence the trait to some extent. If you think about fruit size, right, and strawberry fruit size, we can imagine they did respond to change and became larger. We suspect there were genes that are associated with larger fruit size. Do you think there are other things that influence fruit size that may not be genetic? Probably, right? So you can imagine water availability, nutrient availability, all sorts of environmental conditions. These other environmental conditions outside of genes are going to influence the kind of fruit size you can produce. So a lot of traits that we look at often, there's just not one gene that influences the kind of phenotypes that we see, but it's often genes and environment interact with one another. So we'll be spending a lot of time on that. And this is just kind of an introduction. And then reproductive success is related to the trait. That is, there's some correlation or relationship between the phenotype that an individual has and its reproductive success. That is, how many offspring does it contribute to future generations? In this particular case, we selected those traits. That is, we conferred bigger fruits having higher reproductive success because we only let their seeds found the next generation and killed everything else, okay? So it's analogous, but it was caused by us. But the reproductive success in other situations that are outside of artificial selection, reproductive success is related to the trait and the interaction with the environment. Okay, and we'll be talking about the physical environment, the biotic environment, how all these things influence natural selection. So just to convince you, and we'll be spending the first part of the course is going to be spending a fair amount of time on looking at antibiotic resistance. Did you hear what happened in Nevada? You didn't hear over the weekend? A woman died, and it can be a fairly complicated thing. A woman died, and she was she could not be created by any antibiotic. Okay, so one of the big things that people are looking at right now is sort of super resistance to all antibiotics. And what I'm going to suggest to you is evolutionary biology may have some insights into this problem and process, okay? And there's a whole chapter in the book on evolution in medicine. And I think that it might be helpful. So one of the things we're going to be looking at, there are some practical applications of understanding evolution via natural selection. One of the really most important things is controlling resistance to antibiotics and insecticides. And we'll be spending a fair amount of time on all of these things. But and there are reasons we do it, right? People are sick. We have plants that we care about, right, that are important food crops, and we want to minimize their damage from all of these parasitic things, right? Don't get me wrong, I'm not arguing against any of this. But we set the stage for widespread, rapid resistance from microbes and insects through the processes that we use. And I'll just leave it at that for now, okay? The other thing I think it's important is we can address problems in conservation of biological diversity and issues in medical science by understanding evolutionary biology too. So I'm going to try to use this as some of the key themes that we use throughout the course, and I think hopefully we'll convince you it's an important topic. Okay, this is just an example of how quickly so these are the major classes of antibiotic drugs that occur, starting with penicillin. Then we have these cephalosporins and these carbapenems. I think that's how you pronounce them. Those are the most commonly used ones right now. And what you can see, there's another one that just came out fairly recently, 2000. But the pattern that you see is the dark colors represent the time when they were first introduced, and then the date the first resistance observes is in the lighter color. What's it look like there? What's the pattern that you see on this whole suite of antibiotics that we have used from about 1945 up to about 2000? It doesn't take very long, does it, for antibiotic resistance to evolve through these various bacteria and these sorts of things. Okay. So one of the things we'll be spending some time on is looking at how antibiotic resistance evolves. Why does it evolve so rapidly? And then eventually get to some point where we might talk about, how can we change our pattern and use of antibiotics to minimize the development of antibiotic resistance. And one of the really kind of interesting things is when you think about pharmaceutical companies, when you think about this period in the 60s and 70s up to 90, there was a huge investment in research and development of antibiotics, right? It is amazing how little R&D is going into antibiotics in sort of pharmaceutical companies right now, and people are, I don't want to freak you out, right? But there's some concern about, the development of, and many bacteria are becoming resistant, super resistant to multiple strains of resistance to multiple antibiotics, not just a single antibiotic, okay? And so there's some concern there. Here's an example. This is, I think, tuberculosis, a mycobacterium. Just looking at one of the things we'll be spending a lot of time is looking at the kind of spatial distribution of things, right? And how it varies across space. This is a pretty broad geographic or spatial scale. But this is the percentage of cases of resistance to mycobacteria, to the antibiotic used. I can't remember which one it is, for mycobacterium, the thing that causes tuberculosis. And you can see there are certain regions in Asia that have pretty high levels, greater than 18% of drug-resistant mycobacterium. Others have much lower percentage of cases of mycobacterium resistance and that sort of thing. But of course, the big concern is the spread of resistant strains of these mycobacteria to other areas, and why does this occur? Okay, so let's think about just kind of looking at this. So here you can see high ranges of resistance, not so much in North America, not so much in certain parts of Africa. You also see lots of areas where there's no data, okay? The white areas represent data, so that, doesn't necessarily mean that resistance hasn't evolved in that mycobacterium in those human populations, but they could be. So we're somewhat concerned about these sorts of things. So how does resistance work? How are bacteria different from us? Yep. What's their generation time? 10 minutes, yeah, 30 minutes, depending, right? So bacteria have really rapid generation, really short generation times. How would you describe their population size, even within a single individual, much less a human population or huge, millions and billions, okay? So one of the things that happens under these sorts of situations in which you have really short generation times and huge population size is you can get the really rapid evolution of resistance. Okay. And I'm not talking, there's some other weird things that bacteria do with plasmids that really allow them to confer resistance and spread resistance incredibly fast. Okay. And again, I don't want to I'm not freaking you out, am I? Okay. But anyway, let's just look at the basic process. So let's start at A, time 0, right? C is sometime later. So if you look there are a bunch of, I don't know what those are. They're Campylobacter, some sort of bacteria, right? Inside a host. The ones in kind of yellowish are antibiotic sensitive. The ones in red are antibiotic resistance. How would you describe that ancestral population prior to the introduction of the antibiotic ciprofloxacin? I wish there were more bugs in there, but what's the predominant thing in the population? Yeah, the sensitive ones. They've never been exposed. And there appear to be, and you could talk about, well, why would there be any antibiotic resistance? But let's just assume there's some mutation. Some of them have a mutation that confer resistance. You then, obviously, what happens when you introduce the antibiotic into the environment in B? Death, dead, and which ones died? The sensitive ones, right? And so through this sort of intense, often widespread application of antibiotics, depends on how you do it within populations, how many individuals, how widespread the disease is. This can be, they're better mathematical minds than me that are working on modeling all of this stuff. This is a very complex problem, don't get me wrong, and I'm simplifying it to a great extent. But what this sets up is really rapid selection for antibiotic resistance and the elimination of these sensitive strains, such that eventually what you can see here is you eventually have pretty much 100% antibiotic resistance. And this doesn't take very long, and you can see that in the previous slide. So so what you have here is, this is what we call intense natural selection, right? Very strong selection favoring antibiotic resistance in an organism that has extremely short generation times and huge population sizes, such that there are probably going to be a few rare individuals through various mechanisms that either have the mutation or randomly get the mutation that confer resistance, or they get a plasmid from some other bacteria that has a resistance. Okay, there are lots of ways that bacteria can do this, and we have to deal with it. Okay. I'm just going to introduce you to some, what's that thing there? What's that thing on the right? That's a phylogenetic tree. What is that, when you think about it, what is that, what is a did you have this in biodiversity? Did you spend a lot of time on phylogenetic trees and biodiversity? Kinda? Okay. What's down here? And what's out here? What does that little picture represent? Yeah. So what this is, these are the tips of the phylogenetic tree, right? And what's on the tips? Well, living ones and then an extinct one, okay? And then what are these things down in here? Those are the branches of the phylogenetic tree. And what do they represent? Let's look at, here's Elephas maximus A and B. Do you know where's that elephant found? These are primarily the Asian elephants, okay? And these are the African elephants. And these are probably two different subpopulations or subspecies, okay, throughout Asia. What does that node right there represent? That's the common ancestor to those, we'll just call them two subspecies of Asian elephants, okay? That is deeper in time, right? That at some point, those two subpopulations of Asian elephants diverged, right, from a common ancestor. This branch along here represents, it goes straight, but then it goes this way, represents evolutionary change, right? So from a common ancestor, these diverged, okay? We call those two things, what do we call, you know what we call those two? Those are called sister taxa, and we'll be spending a lot of time on this, okay? Here's another group. Those are the African elephants. Maybe there are two subspecies in Africa, right, that are diverged from a common ancestor. And then what's that node down there represent? Yeah, that's the common ancestor to all three species, including the woolly mammoth. And what happened to the woolly mammoth? They went extinct. Who did that? The suggestion is that was probably a human-induced extinction right after the most recent Pleistocene glaciations, probably overhunting or things like that, and some climate change stuff probably contributed multiply to the extinction of the woolly mammoths. Okay, so that's a little deeper in time, right? So woolly mammoths shared a common ancestor. Is the woolly mammoth more closely related to Asian elephants or African elephants? Asian elephants, okay? So they all are sister taxa at that level. And then down here is the deepest node. That's the ancestral node to all of these things. And we'll be we'll be going through a lot of these things. Okay. I think okay. So one of the things as an evolutionary biologist, as a conservation biologist, as a person who's concerned about diversity, one of the things that people are trying to do is understanding how we use phylogenetic trees to make decisions about which things to conserve. Let me just tell you something. I do not necessarily agree with the book says here or these proponents of this approach, okay? So what you read in a textbook, don't believe it all the time, okay? And there are people who have different views, okay? And again, this is all part of science, that people present an idea, and it may not be what everyone agrees to. Okay, so here's a phylogenetic tree of a group of species at the tips, A through J. And here's a question. There's a group of taxa that share a common ancestor, D, E, F, G, H, I, and J at that node right there, right? And then here's another group of species, B and C, right there. This is a tough one. So which extinction event, if you had to ask for an extinction event, which extinction event, red, which caused the extinction of E, F, and I, or blue, which would cause B and C to go extinct, would prune the phylogenetic tree, and how would this inform which ones you might want to conserve? Would you want to conserve B and C, or would you want to conserve three species, E, F, and I? Anybody want to fathom an answer to that? And again, I would suggest reading the textbook in terms of what they suggest about this, but one of them will prune the tree more significantly and change the shape of the tree than another one. And what these and again, I'm not going to ask you about this on the exam, but one of the things that the people who believe in phylogenetic diversity would say is we want to conserve those species in which the extinction of them would cause a more severe pruning of the phylogenetic tree. Want to take a guess? Which one? B and C? Yep. That's what the phylogenetic diversity people would say. That if we had to make a choice, and maybe we can't save everything, right? that what we would want to do, the extinction event, if B and C went extinct, that would change the shape of that tree and the structure of that tree much more dramatically than E, F, and I going. And the reason is primarily, and we'll be talking a lot more, is this is a fairly deep branch, right? And causes the extinction of a fairly deep branch in a phylogenetic tree and really kind of has a greater effect on phylogenetic diversity. But again, we'll be spending a lot of time on phylogenetic trees and looking at those sorts of things. Now, I'll just leave it. I'm going to do one more thing. So I'm just going to leave it. So when you go to Amazonia, right, you see beautiful things like this, right? There's a tanager, there's a kinglet, there's a, is that a macaw? Yeah. Yeah. Huh? Okay, hyacinth macaw. Okay? So when you look at this, I mean, what you see in Amazonia is this is what's called a hotspot of diversification. We'll be talking a lot about why species diversification accelerated in Amazonia so much, especially in the last 10 to 50,000 years. There's been an intense, recent, what we call recent rapid diversification in Amazonia. You can go to certain parts of Amazonia surrounding the Amazonian basin, a little bit up into the foothills and the mountains, and you go one place, you find a unique species, bunch of unique species of birds. You go 100 kilometers away and you find another suite of completely unique birds. Most of them differ in terms of the coloration. And which do you think has those colors? That one on the left, what sex do you think that bird is? That's a male, okay? So much of the diversification that we see has to do with male coloration. Okay, so let's imagine this is Amazonia, right? Here's another group of birds, maybe in North America, right, that has a deep branch. Guess what the phylogenetic diversity people say? What should we try to save? The blue one. Do you agree with that? I don't. Okay. So anyway, I'm just trying to point, again, this isn't going to be on the exam. I'm just trying to get you, you have to think about the thought process here, right? This is, you're still going to have, you're going to have two species here, and two species here, right? But you're going to lose how many? 6 species, right? Whereas there you're going to lose 1. But the phylogenetic diversity people say, well, that's got that deep branch, it's going to change the tree more dramatically than that if you just let the birds that underwent a recent rapid diversification, let them go extinct. I disagree with that. So my view would be, and this is what most conservation biologists would argue, is they don't agree with the phylogenetic diversity people. They would say, in fact, what we would really want to do is conserve those hot spots of biodiversity that have undergone recent diversification and do as much as we can. Now, it may be that we can't save all 7 Amazonian refuges that we see, but people would argue that that's an important thing to do. Okay, so all I'm really trying to get across is we can use phylogenetic trees to help us understand how diversification occurs, and that might then inform us how we conserve things. Okay, I'm going to stop there, but I'm going to ask you, if you can, to read in your textbook about this example. This is a butterfly that, I'll just set the stage real quickly. This is a butterfly in, is it Samoa? No, wait, jeez, okay. I don't remember. Somewhere in an island in Guam, I think it's Guam or somewhere in the Southeast Pacific. This butterfly was infected by a thing called Wolbachia. It's a protozoan parasite that kills males, okay? And what happened is this Wolbachia got into this butterfly population, and it basically changed the sex ratio to about 99% female and 1% male. And we'll come back and we'll talk about next time is, what do you think happened after that sex ratio changed from an even male-female sex ratio to this very female biased sex ratio? And so we'll talk about some experiments that were done. Okay.